Hello everyone. Welcome to this workshop on molecular phylogeny that is organized by the Life of Retaria. So the goal for today is that we are going to try under 1 hour to go through these points, is to present you the most fundamental basics of molecular phylogenies, not really on how it is made, but how you can do it on your own. And for that, we are going to go through four parts. Going to the basics, then to show you where to find data to do your own molecular phylogenies, then how you can do sequence alignments and infer tree topology on your own, and then the time tree theory and how to do an inference. So there will be 3 speaker or teacher today. We are going to start with myself. I am Raphael Mora. I am a senior scientist at Marum in Germany. I am a specialist of foraminifera evolution. I will show you how to do a topology inference. I will be followed by Miguel Sandin, who is going to present to you the time tree theory. And then he's going to be followed by Mattia Greco, that is going to give you guidelines for a time tree inference. We are going to do a break for question after each of the speakers, so that if there is any point that needs to be clarified, you can ask them, but we are going to just run through the presentation. So each presentation should be between 15 to 20 minutes, so that in total it's going to be an hour. So what you will need yourself to reproduce the exercise at home or doing your own phylogeny to address your own question, is to first find a question you want to address. It's very important that you want to test a hypothesis in particular, and it's really your starting point. And for this workshop, we are going to take the case study of the phylogeny of the Globigerinoides genus. Then what you will need is DNA sequences editing program, such as either CView, Mega, or AliView. There are other options. What are good with this program is that they implement basic functions. They are user interface. It means that you don't need to know how to code. This is programs that function with menu, and you can do all basic analysis on your desktop with that. And these are free. And all other resources that you may need, you are going to find them online. So you don't need to worry about anything else. So really the central part is going to be your question and this sequence editing program. Okay, so let's start with micropaleontology. I assume everyone is familiar with micropaleontology and why micropaleontology is great. But I'm going to go through the most fundamental basic as a start. So with this very large ship, we can go at sea and retrieve sediment cores that are archive of life and climatic history. And from this sediment core, we can retrieve microfossils. So that is fossil assemblages, and in there we are going to find a vast diversity of organisms, and we can interpret biological evolution through sediment time series. What you can get out of it is that you are going to pick this macrofossil here. This is the example of foraminifera from the Danian. And from that, you are going to identify different morphology that you can classify in different morphological species. This is an example from a work from Yale et al. 2015. And from this observation, you can interpret the evolutionary history of this foraminifera. And here I put a figure from the paper from Hayes et al. 2011, where you have an interpretation of the Morozovella genus evolution. So all of that is based on the interpretation of the morphology of foraminifera. That would be the same with radiolarian. And there are two fundamental things where genetic can help micropaleontologists. Is to interrogate what are the morphospecies boundaries, because there is extremely large morphological variability within this morphospecies. And also to ask what are the morphological characters that are phylogenetically informative. In other words, what are the morphological characters that you can truly use to infer this topology and interpret the evolutionary history. So in order to connect the work of the micropaleontologists with molecular biology, we go towards a method that is called barcoding. So the idea of barcoding that you can do only on living foraminifera or radiolarian or any living organism is that you are going to take a species, morphospecies that you have defined with morphological character. You are going to extract the DNA of that organism, and then you are going to sequence a part of the genome that is recording enough variability so that you have a sequence called a barcode that is going to match the morphospecies description, meaning that for every morphospecies that you have defined, you will have one sequence that is going to be particular to that species. In the case of foraminifera, the pargon is a fragment of 1,000 base pair that is located at the end of the small subunit ribosomal DNA that we call the SSUR DNA or 18S. The question is, what is this? So here you have a simple scheme about what you can find at the interior of a cell. Inside the cell, you are going to find various organelles which are performing different functions. And here you see that the ribosomes, they are located on, they are quite small, and they are located on another organelle that is called the rough endoplasmic reticulum. And the ribosome, they are performing a function that is fundamental for the life of an organism. Basically, what it does is that it's going to transform messenger RNA that you can see here into proteins. Basically, what it does is that it translates part of the genome into proteins, and this is something that is fundamental for every organism. Without that organelle, no organism can survive because our cells are producing proteins for our survival. That makes that region of the genome in particular, that is fundamental for survival, is going to have a high selection pressure evolutionarily. It means that it's going to have a rather low evolutionary rate, meaning that this portion of the genome is going to evolve very, very slow. And it's going to be great for phylogenesis with what we call deep nodes. It means that nodes that you are going to go to 100 millions or into the history of the organism, but you should take that with a lot of care because it's highly dependent of the organisms that you are working with. So that is our main source of data for foraminifera and for radiolarian as well. So the question is why molecular data are so practical to study evolution. So the reason is extremely simple, but it's counterintuitive. So you have here these two potential data sets we can work with. You have something that is based on morphology and something that is based with DNA. We, as humans, when we look at this different type of data, we love work with morphology because this is something that we can process much more easily than DNA sequences. And that is true for everyone. However, when you work with computers, for a computer, it's much more difficult to work with morphology and automatize the work, but it's much easier with DNA to work. So what made molecular studies so powerful is just because of that. It's because it's much easier to standardize your work with DNA because you have only 4 nucleotides or base where you are going to find your signal. And to interpret morphology, this is something that is much more difficult, and you cannot do it across, let's say, bacteria, mammals, lizards, and so on, but you could do it with DNA. So it means that the sequence data, they are easier to share and compare, and they're easier to process by your computer. What you are going to do to work with DNA is that you are going to try to produce such kind of object. This is a figure, it's called a phylogenetic tree, where here the dots, they represent the taxa of the lowest rank that we call leaves. By convention, you could say that these are species, but it could be single individuals, or it could be several copies of the same gene, but this is your basic unit into your tree. These, they are going to be connected by branches. And these branches, they are going to be connected by nodes. And the nodes, they are representing an hypothetical last common ancestor between the leaves that are supported by, connected by that node. You can use that to organize your leaves into clade of higher rank. And in order to interpret that tree into the sense of evolution, you are going to place a root on your tree. For that, you are going to need an outgroup. It means it's an organism that is sufficiently different from the other in order to trace what is the last common ancestor between that root and what you call the in-group. And then the order of branching is going to give you the hierarchy between the split, but it's not going to be a measure of time, not yet. And that's what you want to reconstruct. So your DNA sequences, basically what you are going to use them for is to construct a matrix of character. And what you are going to do when you are going to make sequences alignments is that you are going to look at nucleotide position where you have mutation or changes. And this is where you have your phylogenetic signal. And this is what you are looking for. You are looking to that signal that is going to help you to build this tree. And from that, you are going to, yes, generate a topology. So you can work with nucleotides, or you can work with codons, meaning triplets of base coding for different amino acids, and the methods are the same. So as an example, I took this tree from Sierra et al. From 2022. It's a tree rather high rank because here you have all the main clades of foraminifera. Here we are dealing just with benthic foraminifera. And there we have an outgroup, which are the polycystine and the acantharia, which are the other retiae. They are acting as outgroup to place the root. This is your in-group with the globotelamia, monothalamia, and tubotelamia that are the three main clade of foraminifera. Here, this number, they are called branch support. These are measure of the robustness of the nuts, or in other words, how much you can trust these trees. So this phylogenetic tree, they are like in statistics, you are going to get a result. And this branch support, they are here to help you to know how to interpret the data and to know how much you can rely on the tree topology. So when there is a number, and when this number, they are closer to 1 or 100, it means that you have a maximum support. Anything away from that means that your support is low. When you don't have a number, it means that the support is too low to be shown. And it's not shown to avoid to have too much information and to increase the readability of these trees. So that is a typical phylogenetic tree that you will try to reconstruct. Yes, and finally, the substitution per sign that is giving you an idea about the evolutionary distances between the different taxa that you are going to measure only on the horizontal scale, and the vertical scale here is meaningless. So here you read it only horizontally in that case. Okay, so I'm going to our case study. So we have chosen the case study of the Globigerinoides genus. So for those of you who are not familiar with foraminifera, this genus is extremely well known because these are foraminifera that are living into warm waters. and they are major providers of environmental proxies, especially to reconstruct sea surface temperature. So we choose that case because it's not too much. It's an intermediate complexity scenario. We have here 5 morphospecies, and the idea of the paper was to test the species boundary into that genus, and to explore the drivers of evolutionary divergence within the genus. And we have here the paper published in 2019, and all the data are available. So here, this is a figure for the morphospecies occurrence in the fossil record within the genus between the Oligocene, starting at 32 million years, until the Quaternary. And the dark, the black occurrences are those that are solid, and it gives you an appreciation of the relative abundance of these different morphs into the fossil record. And the line connecting the different taxa, they are the interpretation of the fossil record to have the fossil phylogeny that is here summarized. So on that figure, we don't have all the taxa that are here, but basically what we have is G. rubescens being sister to G. tinellus. Then we have G. ruber that is sister to G. elongatus, and Conglobatus that is closer from G. ruber and elongatus than it is from rubescens and tinellus, and this is based on the general appearance of this foraminifera. So now the question is, we want to test this. This is our null hypothesis, and we want to test it with molecular data. So what do? Is that we are going to get sequences. So to generate sequences, what you need to do is to go on the field, collect foraminifera either with a net or with scuba diving, then select the foraminifera one by one, clean them, and then put them into a solution. You see it into that last picture. That is called DNA extraction buffer. And from there, you can amplify the SSU with a technique that is called the PCR. It's not too difficult to do, but you need a lab, you need facilities for that, and you need to be able to go on the field. And the alternative that I really encourage you to do is to check for data availability, meaning that many foraminifera, planktonic foraminifera, and benthic foraminifera, and also radiolarian have been sequenced. And if you want to test specific hypotheses, you can go on NCBI and look for data availability. But this will require that you do manual curation, meaning that you need to select your data and make sure that the data you get are correct and they are really fitting the hypothesis that you want to test. And this is what I'm going to show you. Okay. So here I have the portal that is open called NCBI. This is a massive portal. There are many data that you can retrieve here. So I'm going to show you a path that is linear, but it's a jungle. I'm going to just show you how to go the fastest way to your data. So here you can type simply the name of the taxa that you are interested in, and it's easier to start with species name. So I'm going to start with Globigirinoides elongatus. Okay, here at the nucleotide section. And I'm clicking on search. So what is going to return to you are all the sequences present on NCBI that contain this taxa name. And here you will have this list. I'm going to read the first one: Globigirinoides elongatus, isolate T147, clone DS, cytochrome C oxidase, subunit 1, COX-1 gene, partial CDS, mitochondrial. That's a description of the species, the name of the specimen that have been sequenced, and then the name of the gene that has been sequenced. What I'm going to do is that I'm going to show you how to just retrieve the sequence. Here, I can ask to have between 5 or 200 sequences per page to be listed. You could go to them one by one and retrieve more accurate information, but right now I just want to retrieve a batch of sequences. And here on summary, I'm going to click there. I will have a menu that opens. And here I'm going to go to something called FASTA, that is text. And the FASTA file is a FASTA that is readable by the program I was mentioning before, where here I have the same description, and below I will have the sequence that is here. It's a very simple format. It's just text. You start the sequences here. It gives you the name. Then you have return, then the sequences, and then you go to the next sequences. So it's a format that is very easy to understand. So it takes some time to download everything. You see that it's not finished and it continues. But all you have to do to retrieve these sequences is just to do Control-A, Control-C. And then you would just open a new, create a new text file, my sequences. I can leave it in txt. Open this file and paste it there. And that is readable by CView. Okay. So I already downloaded the file. I'm going to open it here. I open it in CView. So here you see that you have a user interface. So each line here is one sequence. And if I go to the right, I will see each position into the sequences. And here, as you can see, my problem is that it's a mess. And what we want to do is that we want to first select the sequences that are going to be relevant to our study. To do that, I'm going to align the sequences because it's going to allow me visually to see what belongs to the same gene and what does not. So I'm going to go on the menu called Align, that is here. This is the third, starting from the left. I'm going to click here. And then I'm just going to click on Align All. It's going to launch a process of alignment. So here there are 200 sequences, so it takes a bit of time, a few seconds, but it's rather quick considering the complexity of the task at hand. So it's going to take maybe a minute, and it informs you about the progress. So these alignments, when you have more taxa or sequences that are particularly long, you can find the same type of program online, and you can make them with methods that are a bit more accurate and with more calculation power. But for this amount of sequences, it's not something that is needed, and you can run it on your laptop, and it's going to take just a few minutes. It was a bit quicker when I did it earlier today because there was no Zoom session open on my computer. But it should be finished. Yeah, so 1 minute, 18 seconds. Okay, so I make it full. So here I see that my bases are aligned. I see that all my sequences, they are not equal. In length, I mean. Sometimes they start at different base. And then if I go on the right, I'm going to see part where I have mutation here. For example, I have some sequences with a C and other with a T. Here I see one sequences that have a mutation. So here this is maybe, let's say, a sequencing error. Here I see a part that is not perfectly aligned. So it just shows you that the method is not perfect. But here it looks more or less organized everywhere except at the top. because at the top I have a few sequences that do not belong to the SSU. It belongs to the COI, which is another gene that is coding for another function. So here what I would do is just get rid of this, delete the sequence. Then I would have here just the SSU, and then I will select just the sequences that are consistent or that are complete. Okay, and this is the curation process. So I'm going to leave that file. I'm going to open a new one where instead of having 200 sequences just to elongatus, I selected 16, 17 sequences belonging to Conglobatus, Tenellus, Elongatus, Ruber-Pink, Ruber, and Rubescens. That is going to help me to test my fossil topology. And so here it's just 17 sequences. You don't need to do it on 100 of sequences. It's not necessary. And here I'm going to do the same. I'm going to align everything. So you see here that it goes considerably faster, 3 seconds, because we have less sequences. And here is my alignment. So from left to right, you are going to see where are all the mutations between all these species. And this is an alignment that is 1,000 base pair long, which is not too long, but the foraminifera evolve relatively fast. So it means that we have enough phylogenetic signal in order to infer the topology. So yeah, I put my presentation again quickly. So we have these 3 steps. First, retrieve and curate the data. This is what I showed you. Then we align the sequences. So align all. And finally, we are going to infer the tree topology. So I'm going back to CView. I need to reduce a bit. Here I have several menus, and the last one is called trees. Here you are going to have all the different computational methods that are available and implemented in CView. It's not all the computational methods that are in existence. And here I'm not going to explain what they are doing and what are their mathematical bases, but this is something very important when you infer trees, but this is not something we are going to cover today. So here I'm going to PhiML, that is what we call a maximum likelihood method, that are a very standard method to infer topologies. I have this window that opens. You have several options that appear that you can modulate. Again, I'm not going to enter into detail about what all of those are doing, and I'm just going to infer the tree. I click on Run. Again, I have a menu that appears, and that is informing me about the progress of the computer. And here we are going to need to wait another minute, maybe. So 30 seconds. And it's being a bit slow. Yeah, sorry. And here is my tree that has been inferred. So again, I have several options. Important one is called branch support. And here I'm going to have this number that shows me what are the branches that are reliable and those which are not. So 1 is good, 0.95 is acceptable, 0.13 is not. Yeah. I'm going back to my presentation, and that's it. You have your tree. So here, this is a tree that has been edited. I put some color that are linked to questions that are addressed on in the paper regarding the genetic diversity and boundaries of morphospecies. And if we go back to our fossil cladograms, and we compare to the phylogeny, we see that Rubescens and Tenellus are not sister clade. You see that Elongatus and Tenellus here are sister clade on the molecular tree. And also Ruber and Elongatius, they are not sister either, even if they are morphologically extremely similar, because Tenellus is sister with Elongatius. This is what we see here. And this group is sister to Conglobatius. And within the Globigeronides genus, this is Ruber that is actually the most distant from the others. And that was it for how you infer a tree topology. Do you have some? Yes, and last thing, sorry, we have a question mark about the sister relationship for Conglobatius because we see that that branch here is not supported, which means that here this is still an open question. That's our best hypothesis, but that hypothesis is not well supported. So that was it for the first part. Is there any question or point that have not been covered?
Maybe we can leave the questions at the end.
So I see no one raising their hands. Are you fine to continue then to the next step? I interpret that as a yes. So Miguel, I think you are going to enter the part that everyone is really interested about is how do you put dates on these nodes?
Okay, very nice. Can you see the presentation? And can you hear me?
Yeah, perfectly fine.
Yeah, thank you. So, well, good afternoon, everyone. We are moving to calibrating the Retaria molecular clock. My name is Miguel Méndez Sandin. And just a brief recap of what we've seen. Rafael briefly introduced molecular phylogenetics and tree thinking. I will be introducing briefly the molecular clock concept and best practices calibrating the molecular clock, especially when it comes to Retaria. And then Mattia will be showing a case study on calibrating the Globigeronides clock. I'm not going to be talking about models and statistics because, first of all, it's not my expertise, and I don't think it's the interest of the target audience of this workshop. So to begin, as Rafael introduced very nicely, a phylogenetic tree measures accumulated change in rate of nucleotide substitution. So in this example, I have split the tree in the different scale, in different mutation units to understand between us all. And we see that the diversification or the phylogenetic distance between taxa A and taxa B is 5 mutation units. So if we know, thanks to the fossil record or any other source, that the diversification between taxa A and taxa B happened 5 time units, let's say 5 million years ago, under a strict clock model, times are proportional to distances. So we can calibrate the molecular clock and infer that the diversification between taxa A and taxa B happened 5 time units ago. But this scale cannot directly scale across the whole tree, so we start having some uncertainties to be able to accommodate the different phylogenetic rates. And complicating a little bit more the molecular clock, we know that a strict clock model is too simplistic at broad taxonomic scales. For example, here we have a broad phylogenetic tree of Radiolaria, and we see that Nacellaria, it's showing relatively long branches compared to different groups of Radiolaria, especially compared to RADP. Because we have the DNA of all these specimens, they are all alive. So basically, they evolved for the same time since their last common ancestor. The only thing is that they didn't evolve at the same rate. So in order to accommodate different rates at different lineages, people have developed the relaxed clock models that allow different rates at different lineages. So very briefly, the molecular clock is this in a nutshell. So now we can begin to calibrate, and something very important that we have to take into account is our scientific question and our dataset. After this, everything else should follow accordingly. But something probably very important as well is how we want to accommodate uncertainty in our models and in the molecular clock. and how we want to apply our priors, either the speciation of the tree or fossils calibrations. The prior is basically the knowledge that we have and that we want to put in our model. Probably the most used approach for molecular clock is the Bayesian approach, or the Bayesian inference, because it allows the uncertainty that you want for the molecular clock calibration. We can use softwares like BIST or BIST2, MCMC3, Mr. Bayes, or PhyloBayes. This software, they can also use the sequence information, which will allow to calibrate much better the relaxed clock model and allow different rates in different lineages according to the sequence diversity. But of course, these methods take a lot of time. They are very computationally demanding. So if you just want to infer a quick tree at the expense of accuracy, we can go to other approaches like penalized likelihoods using R8S or 3PL. That these methods only take into account the phylogenetic distance, and they are very quick, and they actually should provide surprisingly good results. So if we have our scientific question and our data clear, we have chosen an approach, probably the first thing that I would do to start calibrating a tree is to perform a relative calibration and then compare that tree with your fossils. By a relative calibration, I mean that you run the molecular clock as you want it to be, like with all the model assumptions and everything, except with no fossil calibrations to begin. Like this, you see the relative and natural timing of the tree. Then at this point, you are not interested in absolute dates, but more on the relative timing between the different clades. And now we can map our knowledge of the fossil record and start choosing fossil calibrations that we want to use or not. For example, if the fossil record tells us that clade A diversified 10 units, let's say 80 million years ago, and clade C 5 million years ago, we see that there is something conflicting because clade A seems to be the youngest clade and has relatively low phylogenetic diversity, and clade C diversify is not the youngest to diversify and has a substantial phylogenetic diversity. So there might be something off either in the phylogeny or in the fossil record. Ideally, we would be looking for something like this, basically where the fossil record agrees with the relative calibration, like clade A is estimated to be the youngest node, clade B the oldest, and clade C somewhere in between, but accordingly to the phylogenetic diversity represented in the tree. But unfortunately, most of the times we have something in between where it is actually really hard to make decisions on what node relate to what fossil. So this is why it's very important to understand diversity, like Rafael just mentioned. So our phylogenetic tree refers to our sequences. So basically, this represents one genetic species concept. Most of the times for that sequence, we have a picture. So yet again, a different species concept, which is the morphological species concept. And then we want to integrate the fossils, which most of the times refer to a chronological species concept. So for example, in this schematic tree, we have lineages in black, and then they go extinct when that bar turns gray. So when the fossil record tells us that a node is much older than what we see in the molecular clock, it might be because they are both referring to different diversity. So basically, in that example where the fossil record tells us something much older than our molecular clock, might be because we are also covering extinct lineages that didn't arrive to nowadays. And on the contrary, when the fossil record tells us that a node is much younger than what we see in the fossil record, might be because our phylogeny might be covering a much wider diversity that represented in the fossils. So, okay, we can talk about this for very long, but let's keep moving forward. So imagine that now we know exactly what fossil corresponds to what node. Also, something very important is to choose your prior distributions. That this will depend on the fossil, on how you interpret the fossils, how confident you are on those fossils, and again, on the phylogenetic diversity that we have in our tree. So from my point of view, normal distribution allows the biggest uncertainty in your calibration, because not only you allow older dates, but also younger dates in case you were wrong on your calibration. But if you are very, very confident that that fossil represents that node, you can go for an exponential distribution, a log normal distribution, or a gamma distribution. And then depending on how confident you are, you may play along with the standard deviation or different parameters. So again, just to be redundant, because this is important, how you calibrate the fossil might depend on how you interpret the fossils and how confident you are on those fossils. And most importantly, how those fossils relate to the phylogenetic diversity that we have. And finally, imagine that now we selected our approach, we are confident in our calibration, we can finally understand or interpret the molecular clock, which is probably my favorite part of molecular clock calibration. So when you see the phylogenetic trees, Rafael mentioned about some bootstrap support, that's how much you can trust this node. But now with the molecular clock, we have yet a different level of information, which is timing. And most of the times, the nodes represent the median of a posterior probability that we see based on the assumptions that we gave to the model. And that posterior probability, it looks like this. So we cannot really understand these nodes as absolute values, but more like a statistical distribution where the median value is the node that you see. So now that we understand what the molecular clock output is, we can start exploring the evolutionary history beyond rocks. For example, we know, thanks to the fossil record, that radiolaria was present in the mid-Cambrian for sure, but most likely even in the boundary between the Diacran and the Cambrian. And then the molecular clock allows us to go farther back in time and start reconstructing evolutionary history of even sister lineages that don't leave any fossil record behind, like Acantaria, for example. And lastly, it's very important to assess the priors that we gave, so basically the calibrations that we gave in the tree. And this will also allow us to assess our overall molecular clock, but also those calibrations, and to better understand the fossil record for those calibrations that we provided. For example, for the Nacellaria, we still don't know whether the first diversification of Nacellaria happened with the first Triassic forms or the Devonian forms. And unfortunately, the molecular clock cannot turn towards one side or the other one. So we still don't know what are the first diversification of Nacellaria. Also, it's important to see this prior, to compare this prior probability with our posterior probability to better understand the fossil diversity. For example, for Sphaerozoidae, which is a Colodaria family, we have provided these priors, and then the posterior gave slightly older dates. So meaning that maybe phylogenetically we have a wider diversity that represented in the fossil record, maybe because they were not living fossils at the time during their last common ancestor, or probably because they were naked, they didn't have any skeleton. And lastly, when both the prior probabilities and the posterior probabilities agree very nicely, can mean several things. It could mean that the fossil record really agrees with the molecular clock, or the molecular clock rather really agrees with the fossil record, or that we enforce too much our priors and we obtain the data, the posterior probability from the priors and not from the data itself, like it happens for, in this case, for this specific distribution. This is why it's very important to replicate our molecular clock analysis using different approaches, because like this we can get the best of the different approaches. So lastly, just as a take-home message, molecular dating is an iterative process where you start with a relative calibration and you add or remove fossils as you see fit. Has to be interpreted. It cannot be read literally just like the fossil record. And of course, it's best achieved in interdisciplinary collaboration, because it's really hard that a single person have knowledge on both the fossil record and molecular phylogenetics and molecular comparative approaches. So thank you all, and maybe we'll leave the questions for later on, and I leave the stage to Mattia. Mattia, are you there?
Yes. Can you hear me fine?
Yes.
And can you see my screen too?
Yes, very nice.
Perfect. So, hello everyone. This is the final part of our workshop, and we'll take a closer look to at the case study that applies molecular clock analysis. I am Mattia. I'm a postdoctoral researcher at the Institute of Marine Science in Barcelona, and as I was mentioning, we'll now explore an example that puts a bit into practice some of the concepts that we have been exploring so far. The paper in question, our case study, is the paper that Rafael was mentioning earlier. Is a paper published in 2019 on PLOS ONE that investigates the genus Gobigirinoides, the planktonic foraminiferous genus Gobigirinoides, a genus that is of particular interest because within this genus, morphological diversity and genetic diversity are quite uncorrelated, and as mentioned earlier, the aim of the study was to look at the species boundaries and clarifying the evolutionary divergence of within the genus. For the scope of this workshop, we're gonna focus on the molecular analysis, but the paper contains also morphological analysis with the nice CT scan data. So I encourage you all to read it fully if you're interested also in that part. As Raf mentioned, to start this kind of analysis, we always need the raw material is sequences, is DNA sequences. In our case, for the ribosomal marker that Raf mentioned in his presentation. And for these studies, sequences were produced. New sequences were produced for the species, but also the newly produced sequences were also complemented with data downloaded from NCBI and PFR2, as Raf showed you earlier. And again, from the obtained data set, as you saw before, the sequences could be aligned, and from the alignment, a phylogenetic tree could be produced. You see in this tree, the root is Globoturbolothalita rubescens, and what you see here, you see that within 5 morphospecies, we have 8 different genotypes. Genotype here is the second to last column. And this is again to connect to what also Miguel was saying. There are different units of taxonomy that you can refer to. In this case, genotype is a distinct genetic group within a morphospecies. So now that we have this model of genetic diversity, we can frame our question, which is, when did this diversification occur? So to go to address this question, I put here the method used in the manuscript, and I'll walk you through a bit of the terminology used here and to the workflow that was used for the analysis. What you see here is that the software used for such analysis is BEAST. So I'm going to start from the software itself. BEAST stands for Bayesian Evolution Analysis Sampling Trees, and is basically a collection of different programs that allow you to make Markov chain Monte Carlo analysis of molecular sequences. And it has different programs that allow you to create the parameter file, the input file, run the analysis, but also then analyze the data and visualize the final results. And we're going to go through each of the steps together. So the first part, we start always with the data, with our input, and in our case is the alignment that we saw earlier, how is produced, and this alignment is fed into this Beauty, which is one of the companion software that BEAST has to make this XML file. XML file is basically a text file which contains all the configuration, all the parameters required to BEAST to run. And it has a graphic interface, and within this program, it is possible to set all the parameters, as we were saying, substitution model, the clock model, the priors, and also the Markov chain Monte Carlo settings. So back to what is reported in what we reported in the paper. In our case, our substitution model of choice was the GTR. We selected a relaxed clock model, and we also opted for a Yule model that is particularly suited for species level trees. And for priors, we used the first appearance datum of Conglobatus enternellus, as well as the split between rubescens and the genus Globigerinoides. Importantly, we selected a normal distribution, and we used the standard deviation that reflected the uncertainty of the dating uncertainty of the appearance datum. Finally, for the Markov chain Monte Carlo settings, we selected 10 million generation with a burn-in on the first 1,000, saving then every 1,000th generation. So once we have this parameter set, we can then run Beast, the software itself. And this is how it looks like on the right hand of the slide. All these pictures that you see from Beast are taken from the website, by the way, that provides very good resources and material and tutorials as well for this kind of analysis. So basically, once the Beast program is launched, what it does is that it does little iteratively propose new trees with the changing topology of the tree or length of the branches, and then accepts or rejects this kind of changes based on the posterior probability, and then stores all the samples that accept every n steps. In our case, every 1,000 generation, as reported in the text. Okay. Finally, we get once Beast has run, we can we obtain two important output files. One is the log file with all the posterior samples, and one is the tree files with all the trees that been produced with the different branch lengths and node ages. So we can start, once we have the output, to the post-processing part of the analysis, and this is done with all the other companion softwares that come within Beast. All of them have a graphic interface, as you can see, and this, what you see here, is a plot from Tracer. Here you can analyze and assess your log files, as well as looking at plots, and also at the highest posterior density intervals that from your run, as well as getting the estimated sample size, as you can see here in the table in the graphic interface. Once you can assess that your run was fine, you can then go ahead and produce the so-called maximum credibility tree, and this is done with another companion software of Beast, one of those that I was mentioning earlier in the slide, which is called Tree Annotator. So from here you can set a burn-in for the first trees, so to avoid the noisy part of the output, and then obtain like this a tree that can be then visualized with the Nadal software that is also within among the companion softwares of Beast, which is Fig Tree. Here finally you can obtain the final tree that you can then export for visualization purposes, and you finally can get your final output, your tree, which this is what we obtain in that for the Globigerinoides. We see here the stars for the nodes that are being used for calibrating the tree, and while you see here the uncertainty on the dating from the highest density posterior, higher posterior density, and you can see here that we obtain an early divergence in the Miocene between the Ruber and the other Globigerinoides species, And later in the Miocene, you see then one of the nodes that we use for calibrating, which is the first appearance of Conglobatus, and as well as the split between the Ruber and the Ruber albus genotypes. And later then in the Pliocene, another calibrated node with the first appearance datum of Tenellus, and as well as then a differentiation between the different genotypes of Conglobatus. For then later diversification here for the other genotypes within the Gobigerinoides genes. And yes, that's what I had for this. Thank you very much. If there are questions now, we can.
Thank you very much, Mattia. Maybe what I was thinking is that maybe we can stop the recording and ask questions afterwards. Or maybe it's not possible because Nicole has to close the session to start the registering, right, Nicole? Or am I wrong?
No, I can stop it now.
All right.